Hello everyone, I am Eivind Hennestad and today I'll be guiding you through the Ophys Quickstart live script from the Brain Observatory toolbox for MATLAB. This tutorial illustrates how to access and analyze 2-photon calcium imaging data from the VisualCoding dataset provided by the Allen Brain Observatory. In this session, we'll explore how to retrieve a list of available experimental sessions and filter this list based on specific experimental parameters, examine individual session items, and finally plot the neural activity of individual cells. We will start by listing the available session items in the dataset. To do this, we use the bot.listSessions function. This command gives us an overview of all the experimental sessions that are included in the dataset. The first time you run this function, it might take a bit of time because the session catalog is downloaded and cached from the Allen Brain Observatory repository. The function then returns a detailed table where each row represents a session and includes key information like the session ID and the mouse ID, the experiment type, the acronym of the visual region that was imaged, the age of the mouse, the imaging depth, and the full genotype of the mouse. In the LiveScript interface, you can scroll through this table and explore the dataset. But with over 2,600 sessions available, manually browsing isn't very practical, and this is where filtering becomes essential. Next, let's narrow down the dataset using standard MATLAB table indexing. For example, say we're interested in specific sessions recorded at specific areas of the visual cortex or at certain imaging depths. We can filter the dataset to include only, for example, sessions including the areas of the anterolateral, rostrolateral, or anteromedial areas of primary visual cortex. We can include only sessions where the GCAMP expression is enriched in layer 2, 3 and layer 4 of cortex. Sessions recorded at a specific depth of 175 microns, corresponding to layer 2, 3. Sessions where the eye tracking data is available. And finally, sessions using a specific visual stimuli like drifting, grating, and natural movie stimuli. By applying these filters, we reduce the list of sessions to a manageable size, making it easier to focus on sessions that meet our research criteria. We can now select an individual session for further analysis. In the next example, we select the first session of this table, and we use the function bot.getSessions to retrieve a session object representing the session that we're interested in. The session has an info structure containing all the information that's already available in the table itself. But it also has some other interesting properties. For example, it has the linked items, which is containing a set of cells that are recorded in this session. It also has a linked file values group, and each of the properties in this group corresponds to individual data sets of the session. So you can see that it has the fluorescence timestamps and the fluorescent traces. And these are very interesting because these are actually the recorded neural activity values from this session. We can retrieve data from these properties. And finally, we can view and plot the neural activity of detected cells. So for example, we can get the delta F over F from the fluorescence traces DFF property, resulting in a huge matrix of timestamps or time points times the number of cells. We also get the timestamps themselves, and then we use the plot function to plot the timestamps for the first 10 cells of this session. We see that the neurons have quite different response properties, hinting at the range of different neural cell types and response types to the visual stimuli in this session. Now you have seen how to list sessions, filter them based on experimental parameters, and to visualize neural activity. To learn more, I encourage you to explore the more in-depth tutorials and demo live scripts available. Thank you for listening, and I really hope that this quick overview inspires you to try out the toolbox and to dive deeper into the Allen Brain Observatory datasets.